Okay, life's good. All right. So, like we said, didn't expect to be teaching this morning. So we're winging this thing. Literally have nothing planned. But I think I know where we're at. So, we are on this concept of respiration, right? We mentioned last time that, hey, this connects with the idea of like breathing in a way. Respiration. What's happening in respiration is we're taking some kind of a food source and we're breaking it down chemically, and we're going to ultimately liberate the energy that's in that. So several questions. Of course, where is the energy in the food? It's in the bones. And what molecule is going to be able to come and like break it down without getting destroyed itself?
Enzyme.
An enzyme. Okay? Good. So lots and lots of enzymes, lots and lots of energy. I need to release it so that my cells can use it. What is the energy currency of a cell? It's 3 letters.
ATP.
ATP, adenosine triphosphate. What makes ATP a good, ready source of energy? It's in its structure. It's in its charge and its shape. What is it about ATP that makes it so good? Okay. Well, let me back it up. Where's the energy in ATP? Same answer. It's in the bonds. So think about what ATP looks like. I've got this 5-carbon sugar attached to this nitrogenous base. Right? This is adenosine. Hence the A in ATP. Right? This is the ribose. This is, remember in our actually right now, if I just draw this, it's actually up here. It's the phosphate, right? Phosphate, sugar, nitrogen base. Phosphate, sugar, nitrogen base. If this is ribose, which it is, then what is this? Phosphate, sugar, nitrogen base, and the sugar. When you hear phosphate, sugar, nitrogen base, phosphate, sugar, nitrogen base, what are we talking about? What biomolecules?
Sugar.
Nucleic acids. Okay? This is a monomer of a nucleic acid. Phosphate, sugar, nitrogen base. And then I'd attach another one, phosphate, sugar, nitrogen base. And I do that on and on. Well, if the sugar in this monomer is ribose, what would this become?
Ribonucleic acid.
No? Well, I'm sorry, say it again. Ribonucleic acid, yes, RNA. DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. So if I stacked a bunch of these together, I'd have a polymer of RNA, right? Saying it right. Yeah, I'm saying it fast, and you guys are staring at me like you don't know, and that's okay, it's not good, because we've already tested over this kind of stuff, but this doesn't go away. You better believe that by the end of this semester, when we're doing DNA and RNA and transcription and translation, all that kind of stuff, you're going to be able to, you're not just going to know this, you're going to be drawing this. You're going to draw this yourself. Okay? You're going to have to know this. So right now, we got this adenosine, this ribose, this phosphate. Now, phosphate has a charge, right? Charge of -1. When I add another phosphate to it, what's its charge now? -2. Do 2 like charges like to be near each other? No, they don't. So these things want to push apart. It takes energy to put them together. There's literally energy in this bond. But when I do ATP, it's adenosine triphosphate. So that's 3. It just got a lot harder to put this thing together, which means when I release it, it releases a lot more energy. ATP has more energy than ADP, which is a thing. Adenosine diphosphate. If you run out of ATP, your body will use ADP because there's energy in this bond, but not as much as this. Now once you get down to AMP, adenosine monophosphate, well, there's no energy left. Okay? So this is the energy currency. This is relatively unstable. This thing does not like to be attached here. It freely dissociates and it releases a lot of energy when it does. So your body's taking something like oil that you're eating right now, just that Chick-fil-A sandwich just drenched in oil, and you're like, give me some of that, and maybe one day we'll take another class to learn about how oil is just horrendous and rancid, and they put chemicals in it so that you don't smell. It actually stinks. It's rancid. It's poisoned, basically, or it is poison to you, but they put chemicals in it so that you can't smell that. But we just eat it up, and I was right with you doing that until like June when I got my tonsils out and just had all this free time to study stuff, and I was like, what am I doing to my body? But anyway, and you're taking all that energy and you're putting it into respiration and you're converting it into this so that you can use it. Because you can't just go, oil, and have energy. You've got to convert it into a form that you can use, right? Your car will run on gasoline, but you can't run on gasoline. Try drinking gasoline. You won't be around to tell me how it works. You'll just be dead, right? And conversely, you cannot just go putting Chick-fil-A oil in your car. It's not going to work. Your car will die. Okay? You've got to convert it from one form into another form. That's what's happening in respiration. That's the big picture. Respiration is converting the energy of food into the energy of ATP. Okay? And remember, there are different biomolecules can come into respiration in different ways, different steps. We're going to talk about the model of one type of biomolecule. What's its name? Though I didn't teach you last class. It's called glucose, right? We study glucose. What's the chemical formula of glucose? H12O6, right? So we're going to study glucose. C6H12O6. How do I know that it's a sugar? Two ways. 6, right? No? It didn't do it for me. Two ways. One, it's in the name, ose. Sugars end in ose, right? And the other is, this is a multiple of 6. Maybe that's where you're going, I don't know. But the root, if I were to divide this by the lowest common number, I get CH2O, right? Carbohydrate. Carbohydrate. Okay? That's not going away. That's not going. All these things that we've been talking, they just build. So we got to get them. All right, so we're going to look at glucose. It's a 6-carbon sugar. The first step, no matter what organism you are, is to convert this 6-carbon sugar into
2 different ones, 3 different.
2 different ones, right? Okay. What's that called?
Pyruvates?
They are called pyruvates. What's this process called?
Glycolysis?
Glycolysis. It's not my finest axe that I've ever drawn, but it's not so bad. What's the axe doing? It is splitting the sugar, right? So this is glycolysis. Glycolysis has a series of steps. Why do I do glycolysis?
To get ATP.
To get ATP. How many do I get out net? Net is 2. I put in 2, I get out 4, but since it cost me 2 and I made 4, I put those together and I get a net of 2, right? So glycolysis has a net yield of 2 ATP. I can't spell, I put APT. All right. So it has a net yield of 2 ATP, right? And I'm sure I taught you to kind of do that, right? That's how we always do ATP. So, you got 2 ATP. What else do we have? 2 ATP, 2 pyruvates, and NADH. How many of those do I have? 2. So I have 2 pyruvates, 2 ATPs, and 2 NADHs, right? What do these carry? Yeah, right? Each of these has 2 electrons. There's a lot of energy here. There's a lot of energy still here. There's energy here. But if all I did is glycolysis, this is not a very high yield per unit of carbohydrate, or per glucose, right? Most of my energy is still here in these bonds, and it's here in these electrons. Ideally, we're going to do something with this. Whether or not I can do anything with this to make energy or this to make energy depends on the presence or absence of what molecule? No, oxygen, right? So we can go 1 of 2 steps. We can either go to aerobic respiration, or we can go to anaerobic respiration. The difference is presence or absence of oxygen, right? If I go to aerobic respiration, then I'm going to go through 3 things, right? I'm going to go through this link step. And then I'm going to go through Krebs. And then I'm going to go through electron transport chain. Okay. In my link step, my 2 pyruvates get converted into acetyl coenzyme A. In my Krebs cycle, my 2 acetyl coenzyme A's get converted into CO2 and water. Over here, if I don't have oxygen, now the reason you need oxygen is the final electron receptor at the end of the electron transport chain. I have to have somewhere to put these NADHs, right? By the way, did we do the Krebs cycle last time? Did we get into that? Or is that kind of where we were? Might be, let's assume it's where we were. We'll do that in a minute. Let's come over here, let's talk about the anaerobic side. So if I have oxygen, I'm going to go here. If I do not have oxygen, what am I going to do with my NADHs? What am I going to do with these? On this side, we take our 2 NADHs, and our 2 pyruvates, and we convert them. It either gets converted into lactic acid or into alcohol. Then we're able to liberate our 2 NAD pluses. But there, what is missing? What is there none of over here? Aside from, hey, there's no oxygen, that's why we landed over here. There's no more ATP. With me so far? All right. Why does your body do respiration? To make? To eat. We're going to convert the energy of?
Sorry, what did you say?
We're going to convert the energy that's found in glucose or any other food source, and we're going to convert it into energy in the form of ATP. Where's the energy?
Bonds.
In the bonds. Where's the energy in oil?
Bonds.
In the bonds. In protein.
Bonds.
In DNA.
Bonds.
In sugars.
Bonds.
It's in the bonds. Where's the energy in ATP?
Bonds.
It's in the bonds. Energy is always in the bonds, right? You cannot, your cells cannot use oil. They can use ATP. And so you convert the energy of oil into the energy of ATP. What organisms do respiration? Living? Yes. What do you mean by that? Name some. What else? What else? You think small.
Small?
Yeah, think small.
Cells?
Insect?
Insects, yes. What else? Think non-animals. Things that aren't animals.
Bacteria.
Bacteria do this. Yes. What else?
Fungi.
Yeah. Mushrooms totally do this. Who else does this? We've left out like a whole kingdom of critters.
Plants.
Plants do this. Yes. Is there anything that's alive that doesn't do this? No. Like one of the things of being alive is that you use energy. Right? That's one way that we know that you're alive, and it's one thing that's required to be alive is energy. So every living thing does this. Which one step does every little living thing do? No matter who you are, from the smallest bacteria to the biggest giant, you do which step?
Respiration.
Which step of respiration?
Glycolysis.
Glycolysis. Yeah. Okay. Something like yeast, various bacteria, they don't do necessarily the rest of this. If they live in an anaerobic environment, they don't do aerobic respiration. Bacteria don't have mitochondria. This happens in mitochondria, right? So if you don't have mitochondria, you can't do this. So you're not going to see this in bacteria. It's not an option for them. They can't. Okay? So they're going to have to come over here. So everybody does this, but things that are prokaryotes, no membrane-bound organelles, they can only do this. Things that live in environments where there's no oxygen can only do this. That's their only option. But everybody does this, right? The energy yield here is only 2 ATP. That's not so much. We're going to see soon how much more efficient this is, right? Now, the next question is, why make either lactic acid or alcohol? You make lactic acid. You heard last time? How do you know that you make lactic acid? When do you experience it?
Muscles are sore.
Your muscles are sore. When you've been exercising like crazy and you can't keep up with the oxygen intake that you need, but your body's got it, you keep telling your body, do work, do work, do work, and it's like, I'm trying, but I'm dying here. And so you keep giving it glucose, and it keeps splitting glucose, but I don't have any oxygen to put NADH into, and I'm about to run out of my capacity to split glucose. I got to do something. I got to put these electrons somewhere from my NADH. Where can I put them if I'm not putting them at the end of the electron transport chain to make water? Where can I put them? I got to go this way. If there's no... I want you to think of a factory. Okay? I've never worked in a factory myself or an assembly line or anything like that, but I've seen enough. I think I understand enough of it. There's often conveyor belts. Okay? I have a buddy who works at Walmart, and I know they've got like conveyor belts and stuff, so things go. And if something happens and the conveyor belt stops, or like if the stuff doesn't come off the conveyor belt, well then what starts to happen? It piles up, and then eventually it overflows and like it crashes, and you can't put anything else on it, right? That's what happens here. If there's not oxygen at the end of the electron transport chain, there's nowhere for these electrons to go. They pile up. Well, they back up, and now, okay, well, we got to clear this. Somebody's got to do something. We got to get rid of all these electrons in NADH. NADH, and so it goes this route. It goes and it starts putting those electrons into lactic acid. And that way it frees up NAD plus to go back and keep doing glycolysis. The only reason your body ever does this is because it needs to regenerate NAD plus, so it has somewhere to put the electrons of glycolysis. If I can't put them somewhere, I can't do it. And if I can't do glycolysis, you just die. Right? If I don't have somewhere to put electrons, if I can't do glycolysis, I can't even get to ATP. If I can't get any energy, those cells just die. If enough cells die, then a tissue dies. And if enough tissue dies, then an organ dies. If the wrong organ dies, you die. Actually, frankly, almost any of your organs die, right? You can get rid of, like, what can you get rid of? I guess you can get rid of your gallbladder and a few things like that. But by and large, like, if you have it, you need it. You can't just lose it. So it becomes a very big deal, right? I just told you a lot there. I hope we got it. The reason that we go this route is so we can regenerate this. That is the purpose. What is so special about NAD+?
It stores the electron.
It's where the electrons go, right? To convert it into NADH, and I store 2 electrons. Ideally, I take it to electron transport chain, where they store in oxygen to form water, and I yield a ton of energy. But if I don't have oxygen, then I'll go this route. Some is better than none, right? This is really the big picture of respiration. Do you have any questions at this moment before I show you some videos? No? Nothing about that? It was fuzzy? Okay. Then let me show you some videos. And by the way, any video I show you, where will you find it? On Moodle, right? Any class you miss, where will you find it? So I'm going to erase this. We're going to talk about the electron transport chain. All right, folks. I don't know that I remember these well enough to just freestyle this. Okay. So, I'll give you two things, okay? First of all, let's talk about the mitochondria itself. Okay. Up here is cytoplasm. In here is what we call the matrix. This is what they're calling that means between, intermembrane space. The mitochondria is a membrane-bound organelle. It's actually a double membrane-bound organelle. I've got a membrane here, and I've got a membrane here, and I'm going to use it, right? My glycolysis, it happens out here, and it produces 2 pyruvates, right? And then from there, my 2 pyruvates, they go through another step, right, called the link step. And they produce 2 CO2. And they produce 2 acetyl coenzyme. Well, then these 2 acetyl coenzyme travel into my matrix. And in here, acetyl-CoA gets converted into CO2, right? 4 of them, and they leave. And I've also got a whole lot of NADH and FADH. Got 10 of these. And I got 2 of these. And I'm going to use these. In a second, we're going to draw this, blown up to where I'm really just looking at these molecules. But big picture. Glycolysis takes my glucose, converts it into 2 pyruvates. My link step converts my 2 pyruvates into 2 CO2s and 2 acetyl coenzyme. They travel into the matrix, where the Krebs cycle converts them into 4 CO2s and a whole lot of NADHs and FADH2s. So far, so good. This is the geography of respiration, so to speak. You look like you have questions. Taking it all in. Okay. The geography matters here. Everything, God has literally designed this to work in one precise way. And it is amazing how this works. Ready? So what we're going to do now is we're going to draw an expanded view of like this.
You have 4 minutes for that.
Pardon?
You have 4 minutes for that.
That's all I have. Real quick. Okay, here we go. this is my matrix. This is my intermembrane space. Of course, up here is my mitochondrial. This is cytoplasm out here, right? So this is all phospholipids. Lots and lots of phospholipids here. Hydrophobic, right? Charged things don't like to get through it. So these, because the inside is all hydrophobic, so these H+ ions that I've got all around here that I'm making, they don't flow very well. NADH comes here. And as it does, the energy in NADH is used to pump hydrogen ions. I got lots of them. They're coming off of this. They're coming off of the FADH2. I got hydrogen ions everywhere. Well, I'm coupling the energy from these electrons to pump H+ ions against their concentration gradient. This is active transport. It's one of the things you got to understand, right? So we're actively transporting hydrogen ions against their concentration gradient from one side of the membrane to the other. We're going to build up a serious concentration of hydrogen ions here. The energy for that comes from the electrons in NADH. Okay, and so I'm pumping them, and then my electrons get transferred on down the line. This is called an electron transport chain, right? So we just keep moving electrons, moving electrons, moving electrons, until the electrons ultimately end up here. At this step, I get my FADH2. It comes in at a little bit of a lower step. But its energy is, and here when I get to this step, again, we are pumping hydrogen ions across. We are building a concentration gradient up here. Lots and lots and lots and lots and lots and lots and lots of these. We're forcing them to go where they don't naturally want to go. Diffusion says you want to go from high to low concentration. This is hypertonic. This is hypotonic. They want to flow here. I'm making them go this way against the gradient using electrons. They want to flow back. The only way they can flow back is through ATP synthase. So ATP synthase is going to use their natural concentration gradient to make ATP. Now, these electrons themselves, they're going to end up combining with water, or I'm sorry, with oxygen to produce oxygen plus 4 H+ ions. That's further going to help my concentration gradient. Even the ones that are here, I'm using here to make water. I make 2 H2Os. So I build more of them up here, I take them away down here, I pump them from down here up here. I got a serious concentration gradient. They desperately want to flow down, and as they do, they flow through this beautiful thing called ATP synthase, which takes ATP, or ADP and phosphate, and this thing literally turns. You know, like a turnstile at the grocery store or something like that? Or like at a subway or something like that you walk through and it turns, or like a vending machine and you put a quarter in and you've got to crank it and it turns, right? And you can feel the gears. It's got like however many turns and then it clicks, right? That's how this is. And next time I'm going to make you do this with me. Ready? I mean, literally, we'll be standing up and we'll go 1, 2, 3, 1, 2, 3, and you're going to be like, why is this guy such a dork? But you'll have it in your head, like, oh, it literally, every time 3 of these go through here, it slaps together ATP. It uses the energy, it's like building up energy until finally there's enough, and then it slams that last phosphate into that ADP, converting potential energy into kinetic energy into chemical energy. It's amazing. Have I told you that you are amazing? You're fearfully and wonderfully made. If God didn't make you like this, he's sitting here talking about, oh, and it was like they invented the first thing before you think. Before there was anything, God put this together. It works perfectly. It's incredible. All right. The Lord bless you and keep you. Study. Do your quizzes. If you can ace those quizzes, you can ace this test. All right.
Is it gonna be real like how it was last time?
You know what? Come here. I'll give you a sneak peek. Not long, but sneak peek. So like, can you label these cells? Okay? And I gave you one with these cells, right? Can you label these cells? Can you match? This is right off the thing. Can you match these things to that? And can you tell me off that chart, like, oh, is it in plant cells only? Is it in animal cells only? Is it in both kinds of cells? Is it in bacteria? And then 2 essays. I'm not giving you those. Maybe I should. Kevin left. Jim? He's not here. Nah, I'm not giving you that. All right.
Can we have a little test?
I got to grade one more. Yes, I'll give them to you next class. Okay. I'll give them to you next class. And I'll let you know where your grades stand.
I don't want to see it.
I don't want to say
I'm going to give you, there will be redemption coming.
Thank you. Have a great day.
God bless you, brother. You too.